Hello, my name is Philip Longueuil, and this is my video for the honors option for LB 145 at Michigan State University, taught by Dr. Peter White. Right now, humanity is being ravaged by its worst pandemic since the Spanish flu 100 years ago. As of the making of this video, there have been reportedly over 2.8 million cases, of which around 197,000 have resulted in death. Though this virus has been known colloquially as coronavirus, that is merely the name of the group of related viruses that it belongs to. As much as I would like to talk more about COVID-19, I'm going to instead focus on a more well-studied coronavirus, known as SARS-associated coronavirus, which I will hereafter refer to as SARS-CoV. As its name implies, this particular coronavirus is associated with a disease known as SARS, which stands for Severe Acute Respiratory Syndrome. Much like COVID-19, this virus primarily infects the tissues of the respiratory system, and SARS is characterized by symptoms such as headache, fever, muscle ache, and chills at its onset, followed by shortness of breath, continued fever, and a dry cough after roughly 7 days. The virus is also capable of directly causing pneumonia, as well as causing a secondary bacterial infection that itself can cause pneumonia. SARS-CoV was first identified in February of 2002 in this province of China, and went on to cause an epidemic that is considered to have started in November 2002 and to have ended in July of 2003. There were over 8,000 cases and 774 deaths in 36 different countries. Dr. Carlo Urbani, a member of the World Health Organization and an expert on communicable diseases, was the first to discover the virus. However, as fate would have it, he died of SARS in March 2003 at the age of 46. Now, you may ask, if these are both coronavirus, why is this one so much less devastating than COVID-19? Well, the answer to that may be because COVID-19 is more easily transmissible when symptoms have just started showing, and may in fact be transmissible even before symptoms begin to show at all. Now, let's talk a little bit about just how this virus works. It is thought that SARS-CoV was originally spread to humans from an animal via zoonotic transmission followed by mutation, though which animal in particular, it is not known. The main way in which SARS-CoV is transmitted between humans is when someone who has the virus coughs or sneezes, expelling droplets in the air which others breathe in. It can also be transmitted due to contact with mucous membranes. Like all other viruses, SARS-CoV is not considered to be a living thing because it requires a host cell to reproduce, and is essentially just a giant molecule. This is because it is made up of no more than genetic material surrounded by a protein capsid, followed by a membrane envelope that consists of a phospholipid bilayer. This genetic material consists of single-stranded RNA rather than DNA. As you can see, the nucleocapsid has a helical shape. You may also notice spike proteins that come out of the virus, and this is in fact where coronavirus gets its name, as the spikes are thought to look like a crown, which translate to corona in Latin. These transmembrane proteins are also the means by which the virus gains entry into a cell. They are made up of glycoproteins that bind with a receptor on the host cell that is normally activated by vesicles. At this point, the virus enters into the host cell via fusion of phospholipid bilayers between them, and the virus dumps out its RNA into the cytoplasm. Some of this RNA goes on to be translated directly into proteins, which will form the bodies of the new viruses, and it's possible since the RNA is positive sense. The rest of the RNA undergoes replication in the nucleus. Following this, the now combined proteins and RNA go through the endoplasmic reticulum, where they are given an envelope. Finally, they pass through the Golgi apparatus to undergo any final protein modifications, and then exit the cell via budding in a process known as viral shedding, taking some of the host cell's membrane with them in the process. All that's left now is for the new viruses to travel to neighboring cells and repeat this process. But what can we do to stop this process from occurring in the first place, besides just avoiding exposure to the virus at all? It turns out, not much. As of this recording, there have not been any successful vaccines developed to combat the SARS-CoV virus in humans, though there have been some developed for several different animals. There have been vaccines tested on human subjects, and it was found that they produced the correct antibodies. However, the level of antibodies significantly decreased after 4 weeks, meaning that may only be a short-term solution in this particular trial. Fortunately, there are some ways to mitigate the danger of the pneumonia that is the hallmark symptom of SARS. Ventilators may be used to assist in a patient's breathing if they are struggling to do so by themselves. Antibiotics may serve to combat any secondary bacterial infections, and swelling present in the lungs may be reduced via administration of steroids. The good news is that coronavirus on surfaces is susceptible to a number of agents, including bleach, benzalkonium chloride, ethanol, and isopropyl alcohol. However, keep in mind that the alcohol must be present at 70% or higher concentration in order to be effective against the virus. Oh, and of course there's also soap and water, which reminds me, don't forget to wash your hands. Bye, thanks for watching.